I often have science news segments about ancient species that were discovered preserved in amber, having been enveloped in the yellow gold substance millions of years ago. The amber deposits in Myanmar provide countless discoveries from the ancient world, having encased all manner of various bugs, plant parts, and vertebrate tissues from 99 to 101 million years ago. It just so happens that some paleontologists and biologists from the universities of Göttingen and Helsinki and the American Museum of Natural History in New York were all working together to analyze some amber from Myanmar, and in one of their samples, they found the preserved remains of a curious organism. To quote a press release from the University of Göttingen, they, quote, discovered the oldest slime mold identified to date. The fossil is about 100 million years old and is exquisitely preserved in amber from Myanmar. The results have been published in the journal Scientific Reports, unquote. The press release continues, explaining, quote, slime molds, also called myxomycetes, belong to a group known as the amoebozoa. These are microscopic organisms that live most of the time as single, mobile cells, hidden in the soil or in rotting wood, where they eat bacteria. However, they can join together to form complex, beautiful, and delicate fruiting bodies, which serve to make and spread spores, unquote. Unfortunately, fossils of slime molds, especially their fragile, short-lived fruiting bodies, are super rare. There's only 2 confirmed fossils of slime mold fruiting bodies, and both of these are relatively young, at only 35 to 40 million years old. This newly discovered slime mold, preserved in amber, is 100 million years old. In the sample of amber, alongside the slime mold's fruiting bodies, there were also residues of tree bark and tissues from a lizard. Professor Juko Rikinen from the University of Helsinki said, quote, the fragile fruiting bodies were most likely torn down from the tree bark by a lizard, and the tree bark was also caught in the sticky tree resin, and finally embedded in it together with the reptile, unquote. What they also found in the amber was the slime mold's spores, and curiously, these spores were extremely small. Professor Rikinen also said, quote, we interpret this as evidence of strong environmental selection. It seems that slime molds that spread very small spores using the wind had an advantage, unquote. What they're basically saying is that they could identify this ancient slime mold species as a member of the Stemonitis genus because of their spores, and they argue that their, quote, morphological stasis, unquote, is due to the strong selection for small spores that can be dispersed in the wind. Professor Alexander Schmidt from the University of Göttingen and lead author of the study said, quote, the fossil provides unique insights into the longevity of the ecological adaptations of myxomycetes, unquote. In conclusion, they say, quote, Stemonitis in mid-Cretaceous Burmese amber is by far the oldest record of myxomycetes known to date. The morphological characteristics of the fossil are essentially identical to those seen in extant species of the genus, revealing that all key features of the sporocarps have resisted change for at least the past 100 million years, unquote. This is really cool stuff, because it gives us insight into the evolutionary history of slime molds. These soft organisms, much like the fungus, rarely fossilize, so any little fossil that we can find is often very valuable and can give us a lot of information. But because they're so rare, their evolutionary origins are shrouded in mystery.